Proteins are the next biological molecule we're going to look at. An example of proteins are enzymes that promote specific chemical reactions. Proteins have really specific structural and functional capabilities. Here's a list of kind of basic functions. Some are structural, like keratin, which forms hair, nails, scales, feathers, and horns. Others, like actin and myosin, allow muscles to contract and promote movement. Antibodies work in the immune system and allow for body defense. Albumin is a storage type of protein, and signaling by hormones like insulin is possible with proteins. One of the biggest things proteins are known for is being enzymes, being catalysts. Amylase is an example of an enzyme that's found in saliva that helps break down carbohydrates. So we've talked about already briefly areas where we can find proteins. They make up 20 to 30% of cell mass, and they're found throughout the bodies of organisms, including hair, horns, feathers, nails. But what are proteins? Well, proteins are polymers. They're formed from chains of amino acids. So the subunit of proteins is an amino acid, or you could say the monomer of a protein is an amino acid. Amino acids are made up of a central carbon surrounded by a variable R group, a carboxylic acid group, and an amino group. The only other thing that is there that we haven't talked about is an attached hydrogen. Here in this image, you can see the basic amino acid structure. On the left, we have the amino group with its nitrogen. On the right, the carboxylic acid group that has its carbon double bonded to oxygen. And you can also see the variable R group. That's what is different among each amino acid. It's that variable R group that actually kind of communicates or interacts or gives an amino acid its distinctive properties. Because R groups can be hydrophilic or hydrophobic. They can form disulfide bonds. So they're the interactive part and the variable part of amino acids. So here's a quick look at the amino acid structure again. And then we also have a look at different functional groups that you can find in amino acids. Some amino acids are considered essential amino acids, and for the human body, that means that we have to get them from our diet. The body cannot make it themselves. Here's another look at those functional groups, but here they're highlighted in a little bit better detail. You can see some like glutamic acid and aspartic acid are hydrophilic functional groups. That means in a fully formed protein, they're often not going to be in areas where they interact with water, and that would give that protein certain capabilities and chemical and physical properties. And proteins, just like carbohydrates, are made from dehydration synthesis. When you put two amino acids together, you form a protein peptide bond. So that resulting chain, especially when small, is a peptide. As we keep adding amino acids, you can grow a polypeptide. So dehydration synthesis allows for the formation of a peptide bond, which is a covalent bond between amino acids. And if we wanted to break that, we would just have to add water and perform hydrolysis. But what is most important about proteins is the fact that they are very structurally complex. They have 4 levels of structural complexity. And this is looking at the interaction among amino acids and polypeptides and how the chains twist and fold and connect. It gives proteins a very important 3-dimensional structure. And proteins are all about their structure. You have 4 different levels, so you have primary, secondary, tertiary, and quaternary. The primary level is just the order of amino acids, and that's going to be dictated by DNA. Once those amino acids in a particular order start interacting in a way where they start taking on shapes like an alpha helix or a beta pleated sheet, that's considered secondary structure. When those secondary structures interact to make a more globular protein, that is tertiary structure. And some proteins stop there. Some proteins do not have a quaternary structure. Quaternary structure is when more than one tertiary structure comes together, when you have multiple subunits making up a protein. So primary structure is just the sequence of amino acids. Secondary structure is when those amino acids interact to form shapes. Like here we see a shape which is called a pleated sheet, or sometimes a beta sheet. A spiral or an alpha helix is also possible. Tertiary structure is when we first start to see that globular shape. It's when the secondary structure components start to interact thanks to hydrogen bonding and variable R group interactions. And with quaternary structure, again, we don't see this for all proteins, but it's when multiple polypeptides link together, usually also with hydrogen bonding. So tertiary structure looks slightly globular, and quaternary structure has multiple subunits to create a functional protein. So from order to shapes to interaction by shapes to a globular structure that has many subunits, those are the 4 levels of protein structure. And why look at that? Why is it important? Well, that's because protein function is determined by protein structure. A good example is an enzyme's shape. An enzyme is particular for certain molecules to bind to it. So if its shape is destroyed, it no longer can bind to certain molecules and speed up chemical reactions. That process of unwinding or destroying the structure of a protein is called denaturation. That's when the normal 3-dimensional structure of a protein is destroyed. How can that happen? Well, by thermal means. If you change the temperature, it becomes too hot or too cold. By pH, that becomes too acidic or too basic. If you've ever cooked an egg, you've denatured proteins. You've turned the liquid albumin into a solid albumin, and you can't undo that. So oftentimes denaturation is irreversible, but not always. Next, we're going to be looking at nucleic acids, which are made up of nucleotides, so make sure to continue following along with our look at biological molecules.